My great pleasure to introduce our next speaker, Dr. Daniel Geschwind, who is the director of the UCLA Center for Autism Research and Treatment. He's also the Gordon and Virginia McDonald Distinguished Professor, and he's in the departments of neurology, psychiatry, and human genetics. Dan is a world leader in the genetics of autism and an absolute fabulous colleague. I'm so delighted to have him to talk about advances in ASD genetics.
Thanks. Thank you. I usually say that my mother paid Susan to say that. Maybe this time it was my wife or something. So anyway, it's great to be here. I seem pretty serious, but I have a great sense of humor. And so, as people who know me well know, a ridiculous sense of, so I'm going to, who's aware of Monty Python, the Philosopher Kings? And so as they usually say, and now for something completely different. So I'm going to talk about autism genetics. And it's very, very important. People are saying, why is genetics important? And I'm hopefully going to, by the end of this, you'll understand why it's important. One of the critical reasons is there's several things. One of them is throughout the history of psychiatric diseases, much larger, it's a much larger issue than autism. There's a sense that these disorders, let's talk about depression, schizophrenia, autism, that there's a sense that it's kind of in people's head, or that the diagnoses don't actually have a basis. They're kind of like made up in some way, or, and the advances in biology that you're hearing about from an imaging standpoint, and especially from genetics tells you that from a causal biological perspective, there's a clear biomedical basis for the disorder, and has also kind of closed the circle around to tell us that these disorders, although our diagnostic conceptions change over time and they're fluid, and they're not kind of strict boxes, that they do have meaning that's rooted in biological reality. And that's a critical issue, I think, for the whole field of psychiatry and neuropsychiatry. So what I'm going to talk about today is, I'm going to introduce genetics, the whole paradigm in autism. I'm going to tell you what genes have been identified, not going through them in detail, but kind of what it really means. And then what they mean for clinical practice and treatment. Now, most of that is a promise for clinical practice and treatment. Hasn't been necessarily, it's not going to be a practice for tomorrow. I think Shefali alluded to this in her talk, where having a genetic, we might be able to break the autism, this very diverse syndrome, into components. And I have a disclosure. I consult for SynaptDx that is trying to develop biomarkers for autism. My talk has nothing to do with that, but I always put that up because it's important to have this transparency and kind of sunshine laws. So actually, right now it's an unpaid consulting, but that doesn't matter. They're a company, because I'm just committed to developing biology here. So genes act with the environment during brain development to create a structure. The structure is not a static structure, it's a dynamic structure. So when I talk about what Susan showed you in those images, that's kind of one level of structure, but the structure has multiple levels, from the molecular all the way to the gross anatomy. But the microscopic stuff is turning out to be very interesting and important. And of course, the structure underlies cognitive function. So the brain, like the heart or the kidney, is just an organ and has a structure that comes from that at the top. And so from that standpoint, we can say, the brain is just like any other organ. The difficulty is, and the thing that challenges us, is we know what the heart does. It pumps blood. We actually know what it does at the mechanistic level. And to some level, we could say the same thing with the lungs. I mean, we don't know everything about it, but we have a model for how those organs function and what they're doing, exchanging gases for the lungs and for the heart pumping blood. For the brain, we know that it is the root and the source of our cognition and behavior, but we actually don't have a model for how it produces that. And that creates difficulty. And at some level, and this may seem a little kind of perpendicular to what I'm going to talk about, but that disorders like autism actually, I think, are going to teach us more about the brain than we can actually bring to the brain right now. Because if we can understand how these genes lead to dysfunction in cognitive and behavioral features that are common across humans and animals in some ways, across the animal kingdom, which we're part of, then I think we're going to learn a lot about how the brain works in a way. So does that make sense in some way? And so it's why, but we can start with this notion of development in genes to understand autism. And the model is very simple. You have genes, and if you have a mutation, it gives you a causal anchor. Behavior doesn't cause a mutation. The mutation leads to the organ development. So something happens to affect cerebral structure and function, leading to the autism spectrum condition. And again, the notion here is that autism is not etiologically defined. It's a syndrome that we define based on getting history from the patient and observing them. So we observe cognition and behavior, we talk to the family and get a history, and then we decide that it fits into this box of autism. So it's not etiologically defined. An example would be, again, a biomedical example, maybe that's worth thinking about. There are two of them that we could think about that both shed light. None of these are perfect metaphors, by the way, but I think we can think about them. Is pneumonia, right? It used to be, or fever. Fever is not a disease, right? It's a syndrome, and in some ways that's related to high temperature. Now, autism is more complicated than that, but you know that multiple things can cause fever, just like multiple things can cause pneumonia. What makes it different, though, in pneumonia is that we can identify the bacteria that's causing it and say you have a Klebsiella pneumonia, and we can treat it specifically. And so that becomes a disease then, because it has some specificity rather than being a broad syndrome in which multiple etiologies fit. That's what we're trying to do for autism in a way. So because autism comes from this organ that is the brain, we then realize one of the things, and Susan and Shefali both talked about this, that there are specific things that go awry in the brain in folks with autism, and it varies, but it has to involve social cognition and repetitive restrictive behavior. So there's certain parts of the brain. And so we knew for decades that even before we knew how heritable autism was, that brain structure and function itself is heritable. And so just like height and weight and other things, it's complex, but it's heritable. So genetics is a toolbox, gives us a set of tools to understand how the structure and function goes awry. It's just a set of approach. So I hope that gives kind of some of the foundation. Now, when we look at a patient, or whatever we're looking at in medicine or psychology, we call what we look at a phenotype. A phenotype, from a geneticist standpoint, is anything we can measure. So it'd be your height, your weight, your IQ. Now, whether these have any value is a different issue. But phenotype, as you know, is not just genetic. So here are two identical twin teddy bears, and they really were identical twins. And this is a real-life example. My colleague Nelson Freimer gave these teddy bears to his two young sons when they were very little. One of these teddy bears was loved, and that's the teddy bear all the way over. You can see that teddy bear. And the other one was left in the closet. And, of course, these had the same genetic material, but the environment shaped them differently. And so, it's a funny example, I know, but it just makes the point that I'm not ignoring environment at all. It's just that the environment is extremely complex, and understanding causality using environmental epidemiology or other things is extremely difficult. In fact, the whole field doesn't identify causality, identifies correlations. And so we can start with genetics to give us a causal anchor, and then hopefully we'll understand environmental things, because we're going to be dependent on the environment for treatment of autism. What do we do behaviorally and cognitively? We're altering the environment. The brain is plastic. What do we do with medications? We're changing the environment locally by giving people medication. So in no way, there's often this thing, genes are the environment. No, it's not genes are the environment. Genes in the environment, they work together. We use the genes because they give us a really causal anchor to understand mechanism. And I'm going to explain that right now. So you can find genes. You can then genetically engineer animals. You can make cell models, we call in vitro, like in a petri dish or something. We can also look at postmortem tissue. Now for autism, that's very difficult. The average autistic child has a normal lifespan, so they're not going to die. And men, I don't know, what's our average lifespan now? Somewhere late 70s. If you're married, it's a little bit longer. But it's in the, and I don't mean to, this actually poses it really frames autism as a huge public health challenge because we have children who are going to, grow up to be, adults and, and older adults and stuff. And so it really is an issue. So I don't mean to make light of that, but I do say, postmortem tissue for conditions like Alzheimer's disease and schizophrenia is much easier to come by. It's generally older patients. But, all of this is focused towards developing therapeutics. So we can identify genes, we can study the genes in human patients, and Susan gave you some examples of that. We can actually genetically engineer mice now quite easily. We can, again, look at the mice and say, is what we're seeing in the mice similar to what's seen in patients? And now, with stem cells, and I'll just talk about that briefly, we can put the genetic mutations into stem cells and actually have a human in a dish model, which has strengths and weaknesses, but allows us to do that. So aside from that, pushing the therapeutics forward, in other words, having a way to start to understand how a gene mutation affects brain function and structure in an animal model or in a dish, this is the kind of more clinical side of it. So one kind of mutation is like Down syndrome, right? The parents don't have Down syndrome. That's called a de novo or new mutation that occurs in the germline. So you could contrast new versus heritable and think about recurrence risk. What do I mean by that? So if you have a child with Down syndrome, your risk for having another child with Down syndrome is the population risk, adjusted to your age and all of those things. You don't have 100-fold risk over the population if you have one child. It's because it was a new mutation. So those kinds of mutations actually contribute to autism, not Down syndrome per se. Autism is seen in about 2 to 5% of cases of Down syndrome, so it's slightly increased, but it's not a huge risk. But those kinds of mutations, those de novo mutations, contribute to autism. So how it's inherited can be very important in terms of counseling. Is there consanguinity? That may not be a huge problem in this country, but in other areas it really is a problem. Knowing the genetic base of the disorder has significant implications for prevention, perhaps, right? If we understood some of the genes, we could understand how we could modify things environmentally. The mechanism of mutation may be preventable, right? We live in an environment with a lot of mutagens. I'm not talking about mercury, definitely not. But I'm talking about air travel that we all do. That's, X-rays, things that, indoor air pollution, your exposure to dry cleaning solvents when you bring home your stuff. All of that stuff that we kind of create is stuff that we're all exposed to that's pervasive. They're all mutagens to some level, even though at very low level. So we have to think about that. And then the other issue is, maybe if we can do genetic screening or identify somebody early before, as Ted was showing, it can be a little tricky in the first year of life, but we might be able to identify people who are at very high risk. So far, we have very few mutations, and I'm going to discuss this, but I want to make this point several times. There are very few mutations that are specific to autism, where if somebody has it, I can say, at age 6 months, they have this, they have like 100% chance of getting autism. What I can say is they have a 30-fold increased risk over the general population, something like that, which is enormous. And that would be worth intervening for, no question. So we can identify those things. So our hope is that by working together with these infant SIB studies, with the kind of biomarker work that Shefali and Mirella and Susan are doing, that we can kind of create, panels where we can identify people at risk and then intervene. So the evidence that autism has a genetic origin comes from many sources. It really shouldn't be necessary for me to go over this now, because now, 5, 6 years ago, we had no genes. Now we have over 100 genes. So we know autism has a genetic origin because we can take children with idiopathic autism, sequence their genome, and identify a causal mutation in 10 to 15% right now. But the twin studies for decades, since the 70s, have shown heritability average around 70%. It's an estimate. Could be 55%, could be 80%. It's just a population estimate of heritability. Family studies show that the risk to a sibling is about 20-fold the general population. And again, it depends on sex and all kinds of other issues, but that's the average risk. So that tells you that there's a familial factor. But the real point is that many genetic syndromes or known chromosomal disorders for decades have been known to be associated with autism. So we already knew from the 80s, from Chris Gilbert's work and other people, that there were rare Mendelian genetic disorders, that is, Mendelian recessive or dominant, like Mendel described in his Ps, that are inherited. So now, before I get into telling you what we found, I'm going to give you a little terminology. So in humans, we have 23 pairs of chromosomes. They're the 22 autosomes, and then if we're a man, we have an X and a Y, and a woman, 2 X's. In a particular part of a chromosome, the genes are organized, like they're set out, and all of us essentially have 99.9% of the same organization. It's, humans, even chimpanzees are very much, share 99% of this organization. And within a gene, there are all these different things. So there's exon, intron, and SNPs. So the exon, so what I'm showing here in these RS, those are individual, so the DNA is like an alphabet ACTG. And each one of those things is a place where that differs in the population. Every 100 to 300 base pairs out of our 3 billion base pairs, so there are millions of places where we all differ from each other, but that we differ in a way that's common, shared across our genomes. So that most of these SNPs here are seen in more than 1% of the population. So that means in here we have 2 or 3 people, at least, with each of these variations. And then the gene, this is called PDE8B gene, you can see in the blue. The box part is what's called the exons. In between there is called the intron. Exon codes for protein. So what happens is a gene is a big, big thing, and it's coded into RNA and then into protein. Little pieces of the gene are actually all that's needed to code for the protein. So only 4% of the genome codes for protein. The other 96% is this intronic region. But most of genetic variation occurs in the intronic region, and we still don't know what a lot of it does. So again, this is just showing what a SNP might be. This is the alphabet, like just taking a little piece of the DNA, and there's been a change where the underlying base changes the alphabet from a C to a T. And then there's a complementary strand. So if you have a T, you have an A on the other strand. If you have a C, you have a G. So when we talk about genetic polymorphisms, we're talking about things that are common in the population, more than 1%. When I talk about a mutation, I'm talking about something that's very rare, less than 1%. It's arbitrary, whether it's 1% or 0.9%, but those are the definitions. So when you hear people talk about mutations, they should be talking about things that are very rare. But we have a lot of genetic diversity. We have a lot of things. We have millions, 6 million at least, that are present in more than 1% of us, but that vary between us depending on our ethnic origins. But okay? So that means that we all are carrying some of the risk, but it's not enough to cause autism. You probably need 5 or 10 or 20 of those risk factors. But then about maybe the other half of autism is due to probably rare mutations of various types. So it's really a very, very complex phenomenon. We can detect those rare mutations now with current genetic testing, and I'm going to say that over and over again. So what do those mutations look like? So one type is a SNP, a change in a single base pair. And I told you that 99.9% of our genomes are identical. It's less, 1 in 1,000 we have a SNP. But because our genomes are 3 billion base pairs, that means we have millions of SNPs, these polymorphisms that make us different. What we found out now almost 10 years ago is that it's not just that kind of variation that occurs. I said we had 2 copies of every chromosome. It turns out that in 4% of our genome on average, let's just call it 5%, just for roundness, we don't have 2 copies. We have 1 copy or 3, sometimes 4. And that occurs in all of us, and it's called copy number variation, where large swaths of a particular chromosome, sometimes containing many genes, are actually duplicated or lost. And that's a kind of new idea, and it occurs, again, most of that is benign. It's not really causing a disease in us because we have it. But that turns out to be very important in autism. So we're rich in copy number variation. About 4% of our genome is variant. We don't have 1 copy. We don't have the one from mom and dad. Little piece has been deleted or duplicated. And so this is just showing what that might look like. If you're just reading along the DNA, there are a bunch of genes there. You might have a deletion of here, the D, E, and F, and G are deleted. You can have a duplication, or you can have an inversion of the region, and usually with an inversion, there's small loss of material on the ends. But this is very common. Most of it is benign, but some cause disease. They cause disease when they're large, when they're de novo, that is not present in the parents, like a Down syndrome. In fact, a Down syndrome is a whole chromosome copy number variation. So visible, things you can see under the microscope, aren't really a major part of autism. But these copy number variations, which are smaller, usually smaller than 5 or 10 million base pairs, which can be seen under a microscope, are actually common, probably cause 5 or 10% of autism. And this is just showing an area that's duplicated in autism. This is one of the most common duplications. It's 15q1113. It's one of the most common and most specific to autism. It's just one of the forms. So the first study was published, again, about 7 or 8 years ago. It said that if you had a family history of autism, the risk of copy number variation was lower, de novo copy number variation, but it was about 10% in families that had 1 child with autism. That's simplex. Again, these are just the numbers, and it's basically telling us that, somewhere between 5 and 10% of cases of autism can be diagnosed, the cause of their autism, using one of these microarrays. It's going to tell us what copy number variation there is. And this is just now a new list of some of these disorders. So you can see we're now calling disorders based on the region that's deleted. So a 22q13 deletion, or 22q11 deletion, or a 16p, etc. Some of these are mutations. Like this, I'm going to talk about this a little bit later. This is a recessive mutation found in the Amish in a gene called CNTNAP2. That turns out to be the most common neurodevelopmental disorder in the Amish. It's a recessive disease, which means that you have to get a mutated copy from your mother and father, and recessive diseases are way increased in populations where there's a lot of intermarriage, like the Amish. And so it turns out it's kind of tragic that this was recognized about 8 years ago as a separate disorder, and it's been increasing massively in their population. The point is that we can find these genetic causes, Fragile X, by screening and the copy number variation in 10 to 20% of cases. So it's pretty remarkable. So this is indicated as a genetic test. What's happened since this revolution started in 2007. In 2010-11, it became possible to do sequencing of the genome. And this is showing Moore's law, which is from Gordon Moore of Intel, where he predicted in the 60s that the density where we could pack transistors onto a chip would double every 18 months. That's shown in the white line. The yellow line is showing the cost per base pair in genome sequencing. So this exponential drop is like, exponential squared, right? We're getting down to a point where a genome today, realistically, in and out the door is about $2,500. And the exome sequencing is even cheaper. For 2.5 years, we've been doing exome sequencing here as a clinical test in the medical center. Over 1,000 patients have gone through exome sequencing. The diagnostic yield is 25% roughly. So if you think about that, there's no single diagnostic test, MRI, blood test, or anything that gives you that high a yield. This is a very powerful way to diagnose people. Now, how does that help you now? Well, one thing it does is it ends the diagnostic odyssey, trying to find out what caused my whatever. And it's not just for autism, it's for a large number of developmental conditions, even adult conditions, neurologic conditions, we're sequencing people and have a very high yield. But what this allowed is a scientific breakthrough, really, in 2012. So 4 groups, including ours, collaborating with other groups, published the results of sequencing the coding genome, that protein part, the 5% of the genome that codes for proteins, in about 1,000 patients. And about 9 mutations were found that were very, very high probability. The 6 most frequent genes, though, account for less than 1% of the cases. So what we have incredibly heterogeneous. We estimate, based on that more than 500 genes will cause de novo mutations. We can estimate, we can kind of say, well, we've sequenced 1,000 people. When we do 10,000, we'll have this much. And actually, so now 2,700 have been sequenced. Now there are over 100 high probability risk genes. And the estimate is between 500 and 1,000 genes. That's just de novo exome genes. There was also a strong effect of paternal age, that is the age of the father but not the mother. So this is from one paper. It shows the de novo events were 4 times more likely to come from the father than the mother. That's one thing. And there's an effect where this is just showing paternal age in months, that the number of de novo coding mutations is going up as paternal age increases. How big is the effect? We estimate that the difference between somebody it's about 1.3-fold per decade. So over 30 years, it's about 3-fold. That means that somebody who's in their 50s has a 3 times increased risk. That's a big risk number, but it certainly does not account for a large proportion of autism, right? A 3-fold risk is big. It's kind of equivalent to the overall risk that you might get from a very difficult birth or birth hypoxia or something like that, like an environmental effect. And we don't know what's causing that exactly, but it's known that sperm from older men has more mutations. So, that's been known for decades. So that's likely what's happening. So that's one factor. That's one place where genetics actually helped us understand, wow, there's a gene environment effect here. So just to go over, for those who are the aficionados, for those who don't care, close your eyes for a minute, or close your ears, whatever. So that's showing a de novo mutation. So you have a child with autism, parents don't have the mutation, it occurs in the germline somewhere. That's not inherited. So this is a listing of some of those genes. There are now probably 100 of these that have been identified. There's a recessive, that is both parents, neither of them has the disorder because you need to have 2 copies. Each of them is harboring 1 copy. With 1 copy, you're perfectly fine. But if you have 2 copies, it essentially knocks out that gene. That's something like Tay-Sachs, right? And so there are a bunch of disorders like that. And then autosomal dominant is something where you just get it from 1 parent. Then there's X-linked, that's like Fragile X. Fragile X actually accounts for about 1% of autism. If we have 100 boys with autism, maybe 1 or 2% are going to have Fragile X. And even Duchenne's muscular dystrophy, it's called muscular dystrophy, but there is cognitive phenotypes, impairment associated with that, and a significant percentage will have an autism spectrum disorder. It's not half or anything like that, but it is a contribution. Then we have the complex genetics. Remember I was telling you about all the polymorphisms? What's a polymorphism? Something present in more than 1%. We all share it. It's part of our heritage as humans. We get it from our ancestors. And so the mother and father have a bunch of these. None of them are affected, but it kind of adds up in the patient. We've had much less luck in detecting those so far because the studies, like in schizophrenia, when they studied 5,000 patients, they had 13 loci. We haven't even gotten to 5,000 patients yet. When they went to 50,000, they now have 108 different genes. So we need large sample sizes to be able to detect that additive risk. And in autism, we're under 5,000 still. But there are a few that have been identified, and Susan talked about the MET risk allele earlier. So genetic testing can currently identify about 20% of mutations contributing to autism. So what we do is we do like a Fragile X test if it's a boy. You know, there are very rare cases where a child has a very large head. Well, there's certain mutations that are more frequent than that. So we look at the child and the phenotype, decide on the genetic testing, but it almost always will be clinical microarray, whether it's a boy or girl, Fragile X if it's a boy, and then if all that's negative, exome sequencing is indicated. It's not like, so if you want like state-of-the-art medicine, this is not like optional medicine. So, you know, is this, like there's no ifs, ands, or buts about it. That's what it is. The issue is that this is not being done in most places. Part of that is because the people who are taking care of autistic kids at a lot of places aren't really part of the biomedical establishment. And if they are, most doctors don't understand genetics. That's just, and that's going to change. You know, the whole new breed is going to have to understand this because we're going to be all getting our genome sequenced, and somebody's going to have to figure out what's going on. And you're going to go to your doctor and say, what does this mean? And they can't go, I don't know. So, I mean, you know. But if they're honest with you, this is what most doctors would say. So anyway, I don't mean to make light of it, but it's really a serious issue. So when you read about precision medicine and genomic medicine, this is what it is. And the hope is that by identifying causes, we'll be able to begin to group patients into kinds who will respond to specific types of therapy or help us prognosticate, environmental factors, etc. It's going to be very helpful. I want to take a brief digression because you're going to read in the newspapers occasionally some craziness, right? Like we found the biomarkers for autism or something, and I want to go back to something. So we want genetic biomarkers. We want to be able to say this child has a 10 or 20-fold risk. So the point is, if we do an exome sequencing, we can identify these very rare variants. The question is, can we predict the diagnosis of autism using kind of common variation, this kind of additive risk that I talked to you about? And so there was a paper in 2012 where folks in Australia developed a genetic test. And so we knew this was theoretically impossible because of the lack of signal, as I mentioned, in the additive genetic risk. So, but their data, they actually had replication and all the stuff in the paper. So we looked at it carefully to see what the issue was. So this is just for the scientists, all that crap up there. But I just want to show you, the red is the autism, the green is relatives, the black is siblings, the blue are the controls. And so what happened is they used controls that were unrelated, and they were able to like separate this. But the point is, the siblings are largely overlapping, and the relatives are as well. It's telling you it already tells you there's something in these families that's a little different from the controls, even in people who don't have autism. Now, you might expect some of that, but that really mucks up a classifier. So we began to think, maybe there's something different about the cases and controls. And it turns out they had chosen their cases and controls from different cohorts and different populations. Now, they had supposedly matched for background. They said they did. So we took their classifier and took a separate independent cohort, and there's the weights of the classifier, and there's the case control comparison, and the R-square is 0. In other words, there's no relationship between their classifier and autism diagnosis in an independent cohort. So now we're wondering, okay, that fits with what we thought, but why is it? And what we did is then we looked at European subpopulations, like Estonia, for example, and Finland and places like that. And it turns out that Estonian allele frequency over their control allele frequency has a correlation of 0.55 rather than the autism classifier. So what they're finding are things that alter that tell you where you're from. And so we could actually take their SNPs and show that we could literally build a map of Europe from the SNPs that they used. And again, it shows their autism risk score doesn't classify autism versus controls, which is the blue and red. These curves are right over each other. But what they do is they separate Great Britain from Finland, from other European populations. And so don't believe what you read in the newspaper. Well, moving back to what we do know, I've told you there are going to be over 500 genes, maybe 1,000, maybe more, that none of them account for more than 1% of autism. If we take even the 6 most common mutations, they don't even add up to 1%. So we have the autisms, this kind of very heterogeneous from an etiologic standpoint condition. So our challenge is we have identified many genes, so that's fantastic. And they provide clear targets, like in cancer, like a BRAF mutation. There are specific BRAF inhibitors, and you go after that in melanoma and in other disorders. Raj knows more than I do on this. So these genes provide targets for mechanistic understanding and therapeutic development. But they highlight extreme heterogeneity, and so the question is, are we going to be treating 500 individual disorders, or is there some kind of convergence? So we've been looking at this, and one way to look at it is to ask, during brain development, and these are all the complicated things that happen during brain development, this is embryonic development before this line at 36 weeks, and do the autism genes occur at any time in any process? And so we looked at this, because we know that most of the diagnosis of autism is occurring before 3 years of age, so let's take a look. And what we found is 2 things. Let's see how, that autism risk genes fall into basically 2 major patterns and are highly non-randomly together. One of them are genes that are involved in early fetal development, switches that turn other genes on and off, and those are the red, yellow. And then, I'm sorry, those are the green ones. And then the red, yellow, and orange are genes that are involved in synaptic function, the connectivity between neurons. Shafali was talking about GABA, glutamate, all those things. The other thing we found is that they're enriched in the lower layers of the cortex. And don't pay attention to all of this, M's and everything, but the point is, this is the cortex. These neurons project down, mostly 90% out of the cortex to the rest of our body. These project between cortical regions and across the corpus callosum between the hemispheres. And we found that the autism genes are more superficial. They're in these layers that are involved in connectivity, which fits with the cognitive neuroscience. Okay, so we know that. How does that lead us then to developing therapeutics? So we can begin to put the genes in these pathways or developmental epochs. We can also begin to understand what biological processes they're involved in. So a bunch of them fall into something where proteins are synthesized based on neuronal activity. Another is things that actually regulate neuronal activity itself. So there are channels that ions move through where the action potential, the electrical signal, moves up and down neurons. And then there are the ways that neurons interconnect, what's called cell adhesion. So we can begin to group these things together. And so this, like in cancer, where cancer, this is again very, very oversimplifying, but you could think of it as part of it is overregulation, poor regulation of cell cycle, right? And so you have to block division. Cancer cells divide or they invade. And so if you can block those, you can get some traction. This is the notion here. We're identifying these pathways. So one thing we can now do is we can take patient cells and we can reprogram them. We can take a skin cell and make it into a stem cell and make a neuron. We can make a heart cell or anything. We did that with Ricardo Dolmetsch when he was at Stanford, and from Timothy syndrome, in which 70% of the kids have autism. And so we can use these stem cells to develop, and to begin. So we can take the individual genetic forms, 15q, 16p, 22q, et cetera, and begin to try to understand the neurobiology and do screening in a dish. And our goal is that we can use these iPSC, the cell models, and mice and other things to kind of integrate this data and understand. And one of the questions that people often have is, are there valid mouse models, right? Can you make a mouse model? So what does that mean? We take a mutation that causes autism in a human, we genetically engineer it in a mouse, and then we ask, what happens to the mouse? How does the brain develop? How do the cells develop? How do they connect? And how does the mouse behave? And so it turns out that people have done this with a number of mutations. And I would say these models are valid in the sense that they represent true causes of autism in humans. And what's so interesting is that when people have looked at these, they found that not only do they get, they can see some abnormalities in neuronal signaling or pathfinding connectivity, but the mice actually, in many cases, display, for a mouse, abnormal social behavior and the presence of mental flexibility problems. They have rigid and repetitive behaviors. So it's really remarkable. And this is beginning to help us. So one of the models, the CNTNAP2, we could knock that out in a mouse, and we got a mouse that had abnormal social behavior and repetitive behavior. We were able to treat the repetitive behavior with the drug risperidone in the mice, which blocks dopamine, but it had no effect on the social behavior. So we went ahead for another few years and screened a bunch of drugs in the mice. Screening in a mouse is slow going. That's why we need the cells in a dish, because we can do a lot of them. The mice, screening one mouse model can take several years. We screened about 8 drugs in this model, and we found one drug, oxytocin, believe it or not, that actually increased and the social behavior. And let me see if I can. These are both 2 cages, both knockout mice who are abnormal. One of them got oxytocin about a half an hour before this. The other one just got a saline solution. Whoops, hold on. That went. So and we're able to show that this is due to brain oxytocin, because we could actually genetically engineer a virus that turns the oxytocin neurons on and show that it showed the same behavioral effect. The problem is it only lasts about 2 hours. And so we can discuss that in more detail later today about what one thinks about this. So getting back to that, that's just one genetic mutation, one drug that works. The question is, does oxytocin only going to work in a subset of cases with specific genetic mutations, et cetera? But we have, again, these convergent pathways that can be targeted by drugs. And so this is why a number of pharmaceutical companies have now moved into this field and are now, they had closed their neuroscience 5-6 years ago, and now they've opened up again because in these psychiatric disorders, just like in cancer, there's now a ground truth. They can begin to go after these things. It's not going to be easy, but at least it gives you something substantive to go after. And so one question is, are we going to have to target, let's say, blocking this gene or one of those genes, or increasing it for each specific patient or grouping of patients? The brown shows different mutations that contribute to autism. So you can see that, well, even if there might only be 10 patients with this mutation, or 10 with this, that if you pull all these together, it might be 2 or 3% of autism. So you could group patients together and then figure out maybe they would respond to a common therapy. That's one idea. Another thing, and this gets back to what Susan and Shefali were presenting earlier, is that maybe like in epilepsy, where we're just, there's been a developmental abnormality and we give something to alter excitability in the child or the adult, we can do something to alter connectivity or the brain circuits at that level. We can alter the balance of excitation and inhibition in the brain by these GABAergic drugs or other drugs like that. And so the jury is still out. Both of these are being kind of considered. And I think, and I think the only way we're going to know is through clinical trials at some level. So I want to end with just this last point. Paul, I can never pronounce his last name. Somebody else please do, because I always kill it. Paul was a neurosurgeon at Stanford who discovered that this is from an editorial he wrote in the New York Times. For young people like me, I'm 36, given a diagnosis of cancer, there aren't many words. Then my health began to improve thanks to a pill that targets a specific genetic mutation. Tied to my cancer, I began to walk without a cane, a tiny drop of hope. Unfortunately, he succumbed to cancer. I mean, cancer is more complex than just a single mutation. It is very heterogeneous, and that's part of what we have to understand. But this drug bought him about 3 years with his kids and stuff. And so, I would say it's a bittersweet kind of, success in a way. It tells us that we have a long way to go. But this kind of thinking is the kind of hope that we have. To be able to develop drugs that we can combine with therapy to improve cognition. So in summary, autism risk is largely genetic. Its etiology is very heterogeneous. Genetic testing is warranted and is a clinical state-of-the-art, state of practice. It's a clinical test. Soon, whole genome sequencing will be a clinical test. And so I hope I've given you a hint of how genetics might provide a route to development of new therapies, and some of this depends upon the use of genetically engineered model systems. I have to thank also the people who did all the work. This is just the happy laboratory, working on autism. So thanks for your attention. 45 minutes.